Located in the Dallas-Fort Worth Metroplex, the University of North Texas is among the largest universities in the U.S. with over 45,000 students, and is one of only a few universities designated as an R1 university by the Carnegie rankings, as well as a minority and a Hispanic serving institution. The Department of Kinesiology, Health Promotion, and Recreation's facilities are housed in 2 adjacent buildings and consist of a large amount of space and equipment designated for research and graduate student teaching. We utilize a shared model approach such that these can be used by multiple faculty or graduate students. We have found that this approach allows us to avoid unnecessary duplication and provides more flexibility based on our ever-changing needs. This also provides for better utilization of our space and allows us to purchase additional equipment to expand our capabilities. We have the facilities and the state-of-the-art equipment to fully support and implement the proposed PhD program at a high level. The individual specialty laboratory spaces in the department can generally be separated into 2 categories: the Applied Physiology Laboratory and the Psychophysiology Laboratory. The Applied Physiology Laboratory includes space for biological sample collection and analysis, as well as exercise testing and intervention and other specialized research. We have multiple stations for phlebotomy, and those are set up so we can do either single-stick blood draws or IV catheters for longer-term infusion or serial blood draws. For body composition, we have a variety of measures, including a DEXA. It's an iDEXA from GE, and we have bioelectrical impedance. Adjacent to our phlebotomy area, we have an immediate blood analysis area that allows us to measure complete blood count and erythrocyte sedimentation rates in whole blood. We have a space dedicated for biopsy, and using sterile techniques, we can obtain muscle and fat samples using both the microbiopsy technique and the traditional Bergstrom technique. Using ELISA or EIA, we can determine the concentration of a host of biomarkers such as hormones in different tissues, including in serum and plasma. The laboratory is equipped with multi-channel pipettes, microplate washers, and microplate readers. Using gel electrophoresis and Western blots, we can determine biomarker concentrations and phosphorylation states in different tissues, including in the muscle. The laboratory is equipped with Biorad mini gel systems, power station, transfer system, and a ChemiDoc imaging system.
So this section of the Applied Physiology Laboratory is dedicated to cardiovascular physiology research. We use a number of experimental techniques that allow us to assess various aspects of cardiovascular regulation, both at rest and during exercise. These include electrocardiogram to monitor heart rhythm, finger photoplethysmography to non-invasively and continuously assess arterial blood pressure, waveform capnography to measure end-tidal oxygen and carbon dioxide tension, transcranial Doppler ultrasonography to measure cerebral blood flow, duplex Doppler ultrasonography to measure peripheral vascular function, and microneurography to directly record sympathetic nerve activity. Collectively, these approaches allow us to comprehensively assess cardiovascular responses during exercise, as well as the cardiovascular adaptations that occur following exercise training. Lastly, this allows us to assess the pathophysiology that might limit exercise performance in some chronic disease states.
In this laboratory, we focus on molecular detection of RNA, specifically messenger RNA, long non-coding RNA, and microRNA. We have optimized workflows for both the isolation of that RNA from a variety of sources, including whole blood, isolated cells, and tissue samples, either skeletal muscle tissue samples or adipose tissue samples. We can then quantify and determine the quality of that RNA for subsequent downstream analysis. Our primary downstream endpoints include real-time PCR and multiplexed RNA detection using the NanoString system. So in this laboratory, we've also optimized a workflow for the multiplex detection of proteins using the Luminex FlexMap 3D system. And in this lab, we actually have 2 systems so that we have redundant processes, and we're able to isolate serum and plasma from blood samples and then subsequently analyze those for a variety of clinical chemistry outcomes. We actually have an entire logistics part of the lab. And in that logistics part of the lab, we have the ability to custom label tubes by printing labels directly on the tubes, eliminating the need for traditional labels. We've also optimized all our sample storage to take advantage of that standard 96-well plate format. So we store our samples in that format, and we also process them in that format. That allows us to use our array of automated pipettes to optimize that sample processing. We have 8 ultra-low freezers, the standard -80 ultra-low freezers for sample storage, and some are dedicated specifically to short-term storage, but others are dedicated to long-time archival storage. Another big part of our cryo storage facility is actually our ability to store samples at -150 as well. A really big part too of what we do is monitoring our samples very closely. So we have a complete inventory system for tracking the location of samples, but we also have remote monitoring on all our freezers. So the cell culture model represents a really great tool to test out some of those interventions and see how they respond. Another big part of what we do is actually being able to isolate out those individual subsets that we need for analysis. And so to accomplish that, the primary tool that we use in our lab is actually a basic separation that removes and pulls out lymphocytes and monocytes from the blood. But then we can use subsequent magnetic sorting to pull out individual cells. So we could pull out a certain subset of T cells, a certain subset of NK cells, a certain subset of NKT cells. We also have the capacity to go in and do some real-time flow cytometry analysis. So we have 2 flow cytometers we use most often in the lab. One is a 10-color image-based flow cytometer. So this is like the combination of a fluorescent microscope and a flow cytometer. And then we have a standard 12-color flow cytometer as well. And one of the big environmental needs of people in Texas, and athletes in particular, is a better understanding of heat-related illnesses and heat-related exposure. We actually have a laboratory set up. We have a space set up, an environmental heat chamber, where we're able to simulate hot, humid environments in a safe way. In this laboratory, we focus on cardiovascular outcomes in humans. And specifically, we focus primarily on running-related outcomes. A lot of our research focuses on runners. However, we also have the capacity to not only look at cyclists and cycling, but we also have ladder climbing modality. The other thing that we use exercise for that we feel is a pretty powerful model system is looking at exercise-induced muscle damage, and we do that using downhill running. As you'll see, we have a very large motorized treadmill.
So the clinical metabolism room is designed to be a space that can be used for assessment of energy metabolism and metabolic health in humans. Much of this space is designed to assess the effects of various nutrition and exercise interventions on resting and postprandial substrate metabolism, as well as metabolic disease risk. In this space, we are set up to measure tissue insulin sensitivity using the euglycemic hyperinsulinemic clamp technique, as well as the hyperglycemic clamp, which can tell us the sensitivity of the beta cells of the pancreas to glucose. We can also do a frequently sampled intravenous glucose tolerance test, as well as oral glucose tolerance tests. And we examine the metabolic and endocrine responses to various types of meal tests or post-exercise responses while participants can rest comfortably. When we draw blood samples, we have the capability of doing immediate analyses for their glucose, so we can have real-time measurements of that, and we can also spin down, process our blood samples and store them until we analyze them for particular hormones and metabolites.
I work in this exercise physiology lab, which is one of many laboratories that make up the Applied Physiology Laboratory. When participants are exercising on our cycloergometer, we use a metabolic system which analyzes the expired air and calculates how much oxygen they took in from the air that they breathed. This oxygen uptake is related to aerobic energy production. We also analyze a drop of blood from our exerciser. Blood lactate levels are related to anaerobic energy production. And for some tests, we use these machines which remove some of the oxygen from the air that the exerciser will breathe. This changes the aerobic and anaerobic responses, and by doing so, helps us achieve our laboratory's goal to learn how to accurately measure both the aerobic and anaerobic contributions in exercise.
We have a lot of equipment that can help us understand the biomechanics of human movement. That includes 2 portable force plates that will analyze forces in 3 dimensions. And since we have 2 of them, we can do some of the research that I like to do, which looks at asymmetry of force production. So if we have 1 foot on each force plate, we can measure that. Force plate measurement is also often coupled with velocity measurement, and we have 2 linear position transducers that can do that as well. Along with that, we have inertial measurement sensors that can help us do a full body 3D motion analysis or of specific segments. Back to the previous research I mentioned, we can look at the forces we produce, and if we produce a force asymmetrically, we can see how that relates to motion of our body asymmetrically. And finally, we have 2 fully equipped power racks that can help us study specific exercises. We can do strength testing, or we can actually do full-on training interventions with them.
We are one of the only colleges in the country that has a float tank. You may have heard this referred to as flotation-restricted environmental stimulation therapy, or just float or float therapy. The tank itself, fiberglass shell, filled with about a foot of water. But what's unique about this water is that it's also filled with 1,500 pounds of Epsom salt. What that salt does is it increases the density of the water, allowing the body to float effortlessly on the surface. This leaves the eyes, nose, and mouth out of the water for breathing naturally. During a session, we also reduce the lights, reduce the sound, and really give the body and mind a chance to have reduced input to the brain. While the float happens independently, on the other side of this space, we have our control room. This is where the investigator can monitor the session. So we can look at things like heart rate variability while the individual is in the float. This space is also used for all of our pre and post measurements.
The lab is an open, multi-purpose space that houses neurophysiological and metabolic instruments, a sound-attenuated recording chamber, exercise equipment, multiple computer workstations, a participant waiting area, and a graduate student workspace. We use psychophysiological methods such as electroencephalography, event-related potentials, electrocardiography, metabolic testing, structured interviews, and questionnaires to identify neural correlates and biomarkers of disease. Specifically, we examine the effects of exercise and nutrition on neurocognitive processing, autonomic function, and mental health in at-risk populations, including individuals with depression, concussions, or obesity. We have a strong relationship with the psychology department, which grants us access to the SONA Systems participant recruitment and management software. Access to research assistants and recruitment tools allows the lab to complete multiple projects throughout the year.
Our programs offer educational options at the undergraduate and graduate levels to those interested in gaining knowledge and training associated with performance success, leadership effectiveness, enhanced motivation, improved physical fitness, and better cognitive and emotional well-being. We conduct collaborative research in various sport and physical activity environments and integrate these findings into educational training and interventions that promote positive physical fitness and mental health. We utilize our space to help our students work on counseling and motivational skills with athletes and other performers.
Upstairs, we have a large conference room, which is generally used for research meetings such as lab meeting and serves as a meeting space for the department as well, an area for completing questionnaires for participants, and a student meeting space. There are 2 offices in the back where we do things like neurological assessments, informed consent, cognitive testing, and interviews. In the main testing space here in the Behavioral Neuroscience Research Lab, we conduct research focused on psychophysiology. We have a staff workstation for data entry and analysis, and our laptops are equipped with software for creating exercise protocols for specialized equipment, such as our recombinant stepper, our K5 metabolic system, and actigraph accelerometers. Our lab is equipped with devices for measuring body composition, including a Sika bioimpedance analysis machine and a wheelchair scale. We have 2 recombinant steppers and an arm ergometer that are used for exercise testing. Specifically, we just completed a HIT training study for people with MS with walking disability. With this equipment, we use the Parvo Metabolics True 1 cart, and we have a brand new Biodex dynamometer that is often used for outcome assessment and studies related to maximal force production and rehabilitation protocols. We have an 86-inch TV that can be used for outcome assessment and protocols during exercise studies, and general storage where all of our devices that we need, such as our K5 metabolic cart, accelerometers, and heart rate monitors, are all safely stored.
My research lab has about 1,500 square feet space for our research and student study. We have multiple physical activity equipment and virtual reality equipment in our lab, including pedometer, accelerometer, and heart rate monitors.
The department has multiple student offices such that each PhD student will have a designated office workstation assigned. The department also controls the use of a substantial amount of exercise space that can be used for research, including 6 basketball courts, 2 weight rooms, a large outdoor field, 2 pools, 4 racquetball courts converted into research space, 3 seminar and meeting rooms. We're in the process of increasing the department research space. We have almost completed the conversion of an additional 4 racquetball courts into research space. UNT is building a large research-only science building that'll be completed within the next few years. In that building, approximately 3,000 square foot is dedicated to clinical research laboratory for the department. This will include multiple rooms for sample collection, processing, physical examinations, interviews, etc., as well as large space for exercise testing and interventions. Furthermore, this building will have additional wet lab space for us to expand the biochemistry capabilities in the department.